Welcome, everyone, to the Potavirus podcast. Welcome back. We're super excited today to be here with Kilian Vogele. He is founder of Invitris. And so this is a company making cell-free phages. And we're going to dive into how that's going because many of us in the phage field have heard about this for a while. We know that rebooting phages from scratch is important for synthetic biology and all these AI-generated phages. We need the physical component to be able to actually bring them to life. And so that's why we really wanted to talk to Kilian about what their company is working on in this space. And I met you, Kilian, I think 8 years ago at random when we were both happened to be in Brussels visiting Jean-Paul Pirnay's phage making facility. And I remember he was like, these are these guys, Kilian and Kieran, I think your co-founder, right?
Kilian.
And yeah, and he was like, oh, they're making like phages in a test tube. And we were just starting Phage Directory at the time, me and Jan. So that was just like such a cool chance meeting. And I think you just like won iGEM or something, but you were just starting out with seeing if this technology could work. So welcome to the podcast. We also have Joe here. And so, yeah, let's kind of dive in. Maybe Kilian, you can like give us a bit of background of how you got into this, like take us back to 2018, or even if it was before.
It was like quite around that time. So actually also I kind of stumbled across phages by chance. So initially, I'm from my background, I'm a physicist and I worked in the area of synthetic biology. And during my PhD, I wanted to build some stuff like some artificial cells. So having some membranes which are growing and dividing. And when I had this, I saw, okay, now I have a like bubbling membrane, and then I lose my genetic material to continue to grow this peptide vesicle. And I was wondering, like, how can I replicate DNA? And there I came basically across phages because then phages inject their DNA into bacteria, replicate their DNA there, and then good to go. And I had this in vitro system, so like the cell-free system you already introduced. And basically I wanted to check, okay, can I do with this cell-free system and a bacteriophage together this DNA replication? And that was kind of the starting point of all the research. I started from there, and it was like not any medical question, or because I was new to this phage field. Actually, you kind of read a bit about what phages are, but you didn't really have a grasp for it. And that was like early 2018 when we got the chance to host like this iGEM team. So basically in our laboratory, we got the chance to host a student team of, it was 16 students who were all working together on one project for roughly a year. And I was part of as a supervisor of that team. And we discussed several projects, and one was also like bacteriophages and making them cell-free. First, we were thinking, rather thinking about making an engineering platform for genome engineering of bacteriophages cell-free, because sometimes it's hard to bring the DNA after you modify it back to the bacteria. I mean, for E. coli, it's kind of easy, but some other like capsule bacteria, that's way harder. And that was kind of the starting point. And then early on, we luckily met Jean-Paul Pirnay at like an opening of a local bio lab in Munich from Jerome and Rüdiger. And there, like, we also pitched him as our idea because, like, I mean, he has so much knowledge and overview of the whole field. And there he told us about like that it's even only great alone just to make the phages cell-free because it has a lot of advantages. Like you don't need the pathogenic bacteria, you have less like impurities and like a lot of other advantages, which we didn't know before because like we usually don't produce phages normally. And therefore we didn't know that this is a problem. Also like not making phages for therapy. I mean, it's a difference if you want to make a phage in your lab for just a stock to store it away, or you really want to make it to give something to patients. And with this interaction with Jean-Paul, like more and more came clear that it's like really interesting to make cell-free bacteriophages for therapeutic use. And then we like also pivoted a bit in our project and really focused on that also to collect some phages, see how many different phages we can make in the cell-free system. Also like larger phages, smaller phages, also against different bacteria strains. And then I think we met shortly after this trip to Boston, where we presented all our results and had like a really good time. Like several days where like the whole student team like presented everything, and yeah, everyone had a good time. And overall, we also were quite successful there. But then after that, we're thinking how we should continue this project, and also got this invitation from Jean-Paul to visit his lab where we met you. And it was kind of our first touch points with like more senior phage scientists. I mean, we were just like starting off and we're happy to make like a plug assay and stuff like that.
Yeah.
And yeah, so it was really, really interesting. And from there, I mean, we then wanted to continue this project. And yeah, also like we're thinking, how should we do that? So there was like because you're like a group of students, also I was also still doing my PhD, and how to continue this was quite, then we had different ideas. And it's like, because some of them were still in their bachelor's, some in the master's. I mean, some of them for sure also wanted to continue. And then we thought like, okay, let's try to make it in a kind of a startup direction.
Cool.
Because that's like, then we still can like try to bundle everything kind of together, still work like a bit in the lab, and also like, doing stuff together to continue this. From that, basically, then Invitris came along some years ago. So.
Yeah. Amazing. Yeah. That's such a cool origin. And also crazy to imagine 16 students like productively working on one project. Like, I don't know how to do that yet. But like, that's so cool that worked out like that. Like, how many of the 16 still work with you?
Like, I think 2 are like still connected to Invitris. Also like, some like, I mean, they also want to do their PhD and everything. And like, I mean, that makes sense to go.
Yeah.
And I mean, time-wise, they're now like slowly finishing their PhDs. So let's see what will be the next step. But yeah, I mean, as I
Yeah.
said, they were somewhere like in the second semester of bachelor, and they were also doing really great job. And we also distributed all the tasks, like some masters teaching the bachelor students, having some teams, and yeah, that worked really nice.
So the company is associated with the university? How are the students, where are they getting their degrees from when they get their degrees?
Yeah. So the nice thing was like my PI was giving us a lot of freedom. So, I mean, he's a synthetic biology group, and kind of we were the only people working with phages there. But as a PhD student in Germany, you can like also teach, like supervise master theses and bachelor theses. And I mean, for sure, they went to their normal courses, so they could finish their studies normally, but they were like working with me as a working student or as a master thesis or some internships. There was some freedom from the university, so that was also quite helpful to continue and stay together as a team.
Their degree comes from the same university yours came from?
Yeah, exactly. So we were all in the same lab of my former PI, Professor Simmel, and the degree was from the Technical University of Munich.
Yeah,
Awesome.
that I sort of thought it was probably like that. I just wanted to make
Yeah.
sure.
Yeah. So I wonder if can we talk about cell-free phage production just from a beginner's point of view for people who are wondering, like probably if they're listening to this, maybe they work with phages, maybe they're just interested in phage therapy, because we dive into that a lot. But what is it, what's happening, and how, obviously you can't tell us all your secrets, but how can you possibly make phages without a cell?
Yeah. So basically for that, we usually so you have 3 components which you need to make a phage cell-free. One component is the information where the phage is coming from, so basically the DNA. So it's like the building plant where everything is coming together. Then you need to have kind of the machines to produce the phage, which is like the ribosome and GNR synthetases and everything, which is a normal bacteria cell, together with some buffer where like the energy mix is coming from, and also like the building blocks. Everything comes together and makes this bacteriophage. So basically to get all these three components for the machine which makes the bacteriophage, so like the ribosomes and everything. We start with like a bacteria culture, so usually like a safe lab strain of some well characterized bacteria, and we let them grow. We remove the cell wall, remove the DNA, so that we get like all the proteins inside the bacteria cell, which are basically if you add some DNA, make new proteins, and doing there some purifications and some tests. And if we get then all these enzyme like proteins out, we have also like a big mixture of all the different components which cell needs to run. Like one, you can imagine like the fuel of the cell is like ATP. But we also have something to like regenerate ATP to let it run longer, which is also important for phages. And adding this together with some proper DNA, you can make any protein of choice. And if you add like a proper phage DNA, you can also make like a complete phage from that. So it's really cool. Like all the stuff which happens in a normal bacteria cell just happens then in a big test tube. So you can imagine like we have taken or like mimicking what's inside a bacteria happen in like a normal plastic tube, which is like having a way bit larger volume, making way more phages in one go. And there everything happens like as a normal phage would do their program, and you just put everything together, store it at a constant temperature, and after 4 to 8 hours, you get your sample out, and your phages are ready to go.
Nice.
Okay.
Yeah, I
And
was.
yeah, go ahead, Joe.
I was curious because many moons ago, I was in a lab that wanted to recreate yeast cell biology processes, and we just grew yeast and put it in liquid nitrogen and put the liquid nitrogen in a blender, and we would just took what came out of that, and it would like transport proteins around the cell in the test tube. I mean, around different organelles of the cell in a test tube. So it sounds like you do something a little bit more complicated than that, but it was fascinating that really worked really well.
Wow.
Yeah, I think like also we also did some stuff on mammalian cells for not phages, but I mean we also do other things. And for bacteria, I mean the most important thing is, especially for phages, you need like a really high quality like cell-free system, because for the phages you need to make a lot of different proteins, and they all have to build together, and they are all in the right stoichiometry. And I mean also loading the DNA into the phage head costs some energy. And as there's so much going on, you need a quite well-functioning system. I think that's really the key. So that's the main tricky part, and also like the details on how you prepare the system. I think that's really the key to make phages, because in the beginning we also had some batches which we couldn't produce any phages, and we were screening like I think for 3-4 years to make like a really well-established method to have this well-expressing cell-free system.
Yeah, I'm really curious, like how general is it to like how many kinds of phages can you make with one system? And like, are you using like the same base host soup for a lot of different phages, like beyond what would be able to infect that host in nature?
So it works quite nicely for like a lot of different phages. So we made RNA phages, like normal Caudoviridales, also so Podo, Sifo, Myovirides, all kind of different kind of shapes, I would say. And that did work quite well. We also were able to make like phages which are not coming from our host. So usually if you say you want to have a phage against bacteria XYZ, the cell-free system can make it work, but I cannot guarantee it for like each single phage. So for example, like the like M13 phage, I think it's used quite often, and it's growing out of this membrane when it's replicating. And for example, this phage is not working in the cell-free system because it needs like the membrane to grow out. And even if you're mimicking some membrane parts, like with some lipid rafts in the cell-free system, it's not working really nicely. But like a bunch of different other phages, for example, work quite nice. And also what's cool is like the closer, our initial host is like the cell-free system derived from that can make a lot of different phages without modification, also against other hosts, like in, for example, Klebsiella or some Pseudomonas species. even if it's like not coming from these bacteria, this initial host. So that's kind of quite a nice thing to see.
Yeah.
For some more further like away host bacteria, I would say, there you have to make some tricks. So there we co-express, for example, some important proteins or factors to make this work. Not all of them are working always out of the box, but you can like with some dedicated searches also find the tricks to make it run. The cool thing in the cell-free system is like, okay, these 4 proteins are missing to make this particular phage from, let's say, stuff or whatever. And you don't put in the purified proteins, but you just put in the DNA encoding for the proteins. So you throw in the phage
Wow.
DNA plus some more DNA you ordered from your favorite DNA vendor, and everything like also then works together, which is quite nicely.
That's really cool. Nice. So I guess, it might illuminate some, like as you try things and see which phages fail, you might kind of like learn a bunch about the biology of what makes a phage assemblable that we probably still don't know about most phages.
So do you know, I don't know, this probably isn't that easy to do, but have you looked, a lot of people are now looking into what genes are expressed, and they have been for a while, what genes are expressed early, late, in the middle of an infection. Have you looked at that? Do you know are you seeing, like when you first introduced the DNA into the gamish, that do the early genes get expressed and then only later the middle and ultimately the later ones, or?
Yeah, so we did exactly some of these experiments like with mass spectrometry. So we threw in the phage DNA, took some samples over the time, and sent them to mass spectrometry, and we really saw that you got like this early, later, middle genes exactly as predicted for this particular phage. Because I mean, for a test case, we take a characterized phage, so we know what to expect. And we could also replicate that quite nicely. Also nice, we were also able to see some prior hypothetical proteins expressed, like that we could show that these hypothetical proteins were somewhere in our soup. We don't know what they're doing, but they were like also expressed. So they're not just hypothetical protein, but they were like, we could confirm the expression of these proteins. What I have to say, maybe it's like for example, like a T7 phage replicates like within 20 minutes in the bacterial cell, or like 18 or something. In the cell-free system, it takes a bit longer till you get the first phages. So like after 3 to 4 hours, and you get like the final titer after 6 hours. So it's also like you have everything is a bit slower because the cell-free system is also 10 times less dense than a bacterial cell. And in a bacterial cell, if you see some videos where like a walking protein, like you see like quite in an empty void, but it's not the case. It's like packed like super densely. And the cell-free system is a little bit less packed, and therefore everything is a bit slower. But therefore you also get like better time resolution if you want to check for early, late, and middle genes, which is also quite nice.
Yeah, so cool. So in terms of the why you might want to make cell-free, like you touched on, obviously the phage therapy case when we want it to be less toxic, so we don't have to use anything pathogenic to make the phage, then we probably don't have to spend so much resources on getting rid of things like endotoxin. What other reasons have you come to find are like reasons to use the cell-free system over the natural system?
Yeah, I think like a lot of points are like having also no prophages, for example. Because sometimes I think it's really hard to purify your phage from the prophage because they're quite so similar, so it's a bit harder. I mean, for endotoxins, you can like use some columns. You hope that you don't lose too much phages, but that's a different story. So like endotoxins, we also have a lower amount. But overall, I think also for the more like industrial perspective or like pharma perspective, it's quite nice to have like one system to make a lot of different phages. Because like just changing the input you have, you get a different phage out, and you don't have to like start from your bacteria again to remove some toxic genes, some unwanted genes to make like a pure production strain. And just having like the system, you can produce it like in a huge batch, like freeze it, just taking a fraction of it out, making your production run for your phage X, then taking another batch out of the freezer, making the batch high, and so on. And therefore, like you also have like a lot of synergetic effects to make hopefully phage therapy like more accessible and also like hopefully easier to use. I mean, there's this concept of a phage beam also from Jean-Paul Pirnay.
I was going to ask you about that.
Yeah, sorry, and I don't know too.
Oh, no, go ahead. Yeah, like Jean-Paul's, all his sci-fi writings and like that phage therapy 2035 paper. I think your technology is like a key part of that coming true. So go ahead, yeah.
So I have a question about the sort of extract, or gamish, as I called it earlier. Yeah. How much bacterial DNA is in there? Is there any effort to reduce the amount of bacterial DNA, or do you have the bacterial DNA in the gamish along with the phage DNA?
So we have like really low trace amounts, and there are some steps included in the process where you get rid of the DNA. So like we checked with some qPCR and got like really low amounts, which were also like fine with like we checked with some German regulatory authorities.
And the regulatory people, maybe Jeff was going to get to that also, but obvious question. But so you have, I guess I don't know this, but have phages that have been made in your method been used therapeutically? And if so, what countries and what have the regulators said about this?
Unfortunately, the cell-free phages are not used yet for the treatment of patients. We were in conversations with the German regulatory, but also with some from people from Belgium. The main thing is like where we're still in discussions, like to show that our phages produced cell-free are basically the same as produced in bacteria, and to run there some tests and to see like if this is elective proof points that it is like that. And then I think there can be some use cases for this cell-free production. But
so
I mean,
go ahead.
Yeah, and the other part is like, I mean, in Belgium, you have like this magister preparation where you have to show afterwards that your phage is really good and clean and everything. But in some, if you want to go more clinical trial setting, you have this GMP production where you have to like produce like your batch of phages like in a quite strict manner, and it like takes a lot of time and expertise. But we are also already in the phase where we have this like tech transfer to like a company who would do like a GMP production of our cell-free system to bring our cell-free phages to patients hopefully soon.
Regulators express that they want any tests to be done on the phages that they don't require to be done on phages that are made the old-fashioned way by bacteria? Or, I mean, I guess what I'm asking is, do they have some additional concern? I mean, I know they haven't done it yet, but in the conversations you've had, have they asked for tests beyond what they asked for the phages that are made sort of the old-fashioned way?
I think just beyond what the conventional phage would be tested against was about like the mutation rate of the single phages. So because like, I mean, the DNA, so some phages also replicate the DNA in the cell-free system. And also like if the mutation rate changes from a conventionally produced phage. We also have some methods to in vitro amplify the DNA. And if you do the calculations with the enzymes and everything, and you know the error rates of your normal phage polymerase, which is rather high compared to like some other proteins usually, it should be lower. But I mean, there we need to also prove this or show this first to people.
Got it. Yeah. And so many different directions it could go. But like, it just sounds so cool that you can add exogenous DNA so easily to start. Like, that would open up a whole window, or just like a whole bunch of possibilities with engineering the phages. And I feel like I didn't originally appreciate that, but like, instead of having to, clone in or like express a plasmid in your host cell, or, try and get that extra DNA to make even a mutation in a phage, like you said, you can just print extra genes, basically, probably get them made by Twist or something, and throw that in. Like, have you explored making phage mutations in that way? Because it seems just like such a nice platform for that.
Yeah, I think like for the cell-free system where we did a lot is like, I mean, can imagine like a phage, like a Lego set, which builds itself together just by putting all the parts on the table, kind of, right? And if you mix in some other parts, like which are similar enough, they also build it into the phage. And in the cell-free system, we are able to like mix a lot of different parts additionally together. So to be more concrete, for example, the tail fibers of the phage, which are like responsible for checking which bacteria they want to attack and which not. You can also mix in like a bunch of different other tail fibers to make some phages which you get out of cell-free, which can attack also like different hosts which were not initially able to. So we were even able to show that we can like cross species with that. I mean, you can do that to a certain extent because, I mean, also the phage genome had to be read by the bacteria you put it in. But like to entering the genome, it was like possible just by adding some additional like piece of DNA for a different tail fiber, and then even cross like species with that. And also like adding a lot of different tail fibers, you can make like a phage, like a larger host range than initially.
So cool.
You can like also genome engineer the phages, of course, which is also working quite nicely because you can use all the usual like in vitro assembly tools and also selection tools, and you don't have the transformation step. So that's like a big plus, I guess. And with that, we were also like able to like genome engineer phages, and we can also do stuff which you can usually not do. So we deleted some essential genes to make a phage which you can produce cell-free, which kills the bacteria but not amplifies.
Nice.
And just like to see if we can. So I mean,
yeah.
And then also work quite nicely. So there you can also like, I think you can use it as a tool for like research to see like what does this gene do and like switching off some of them. But you can also like think about like therapeutic uses or to have like a faster screening tool. So for example, having a phage without a tail fiber and then just adding like all the different like tail fibers in the solution and then checking on a spot assay if you see something or not. So.
Yeah. Related to that, have you tried just adding like genomes from multiple phages and seeing if you get some naturally occurring frankenphages that way, like almost like an Appelman's protocol, but I don't know, more likely to work because they're all just really right in there, and to try and kind of have directed evolution just take place and see what mixes and matches you get?
We tried there a bit around, but like kind of stopped at one point because I think they inhibit each other sometimes. And if the Lego kind of self-assembly Lego blocks are mixing too wrongly, I think then they might inhibit each other. Like if they are orthogonal enough, you get these two separate phage pieces which are not mixing, and then it works also fine. But if they mix too weirdly, often, I mean, if they're close enough for each other, then it works. But then it's like also just exchanging whatever 3 proteins, and then you see the same effect. But if you mix too widely, there's some I guess like there will be too much inhibition of the assembly, or I mean there also have some other like self-inhibiting effects. So
If you sort of suggest, if you were perhaps a simpler experiment, like, many years ago, people isolated T4 phages with TS mutants in various genes. So you could take a very simple experiment of taking two different pieces of DNA, both of which had a different TS mutant. And if you put them in separately, of course, you would just get out things that were temperature sensitive. But you could imagine that if you put them in together into your cell-free extract, if you got recombination, sometimes you would get a piece of DNA that had recombined, so it had the wild type copy of both genes, and you would have a phage that wasn't temperature sensitive. I mean, I guess that might be, to my mind, a simpler way of, because, I mean, I guess there could be incompatibility, but those genomes would only be different from each other at maybe just one base in two different genes. So they'd be very similar, and they should be perfectly homologous, obviously. So the combination should like happen quite readily. I mean, this is how whatever Seymour Benzer did this stuff many years ago. I mean, and sort of all the classic phage stuff. But I mean, I guess I wonder if you just started with that might be an easy bar to clear. And then, then you'd be confident, like, yes, we we get good recombination. Hey, I'm talking too much, but
That's
you get
cool.
the point that it seems like that would be a very simple experiment. Like you take TS gene X of T4 and TS of gene Y and throw both those DNAs in there and you get something out that's not TS.
Yeah. I think that's a really, really nice suggestion. So this we didn't do so far. I think also maybe we would have to make a specialized cell-free system for like homologous recombination, and because it's a little bit suppressed in the system we have usually, because we don't want to have some of the proteins interacting with the DNA doing weird stuff.
Mm.
But yeah, I think it wouldn't be too hard to make a special system like which could do more homologous recombination and then exactly testing this T4.
So cool. So where are you at with in regards to like interaction with the outside world with your system? Like, is it a kit that somebody can buy in a research lab? Or are you going to be supplying phages for a clinical trial? Like, are you going to make a drug? What kind of direction is Invitris wanting to go?
So concerning the kits, so we have with some partner labs, some like kind of collaborations where we supply them with the system. For us, I think it's like always important that we can like work together on their problems. So like, because for sure, like if you get the kit, you would get like also some example phages to test and to see, can you run it in your lab or not? Whatever weird stuff is happening and then experiments are not working out, so that we can at least troubleshoot like with a positive control. But I think what we also want is like learning from what they do and also giving them like feedback or like helping them troubleshoot to come to the solution like kind of more together. I think that's really important for us to like both parties kind of have something from that, because we don't want to just like sell this kit like a like ordinary like commodity. And besides that, we're also like working currently like in the direction of like Jean-Paul's phage beam, as we discussed before.
Yeah.
Maybe I will shortly explain what his
Yeah.
vision is and then what part we try to build.
Yeah.
So the idea is like, oh yeah, I hope I say it correctly. The idea would be something like you have a bacterial infection, you put this into a machine, and after a certain time, you get a phage cocktail out, give it to the patient, and the patient is hopefully healed. I mean, and there are a lot of steps happening in between, like some AI finds out what phage you should use. The phage needs to be produced somehow and then also like purified and given to the patient. And with our cell-free system, I think it's like really good thing to use for the production of the phages within such an automatic pipeline. And for that, we got like an ESC grant where we currently develop like something like that, where you basically put in some DNA and then getting out some phage related particle, and kind of in as automated way as possible. So basically having something like a phage printer, which would be part of this phage beam. There we currently also developing, and there are a lot of moving parts as well. So I mean, you have to have the input materials like the cell-free system, which you also need to have in a certain quality, like this GMP parts, but also like having like a machine which could like put everything together and gives you out the phages. So like kind of a printer. And that's also what we're currently like working on. And hopefully with that, let's say in some years or like in the future, that like a hospital would have this instrument on site. You get like a patient sample, it's like this certain Staph aureus or something. And then you tell this the machine to produce which particle, and then it runs and gives you out something which you can use in the patient. I think that's what we're currently like also developing.
Yeah, so cool. And I feel like the whole AI generated phages, and we interviewed Samuel and Claudia about that work at the ARC Institute. But I think it just dovetails well. And I wonder what you think about it, because I could see that phage beam system involving a component where the bacterial strain has been assessed to be something. And it's like, well, do we use a natural phage genome Or do we make one up right now that should work against it? Either way, your kind of system would theoretically be able to build it, maybe. Like, do you think that piece, like the AI generation of a new phage, is required for this type of system? Or like, how much of a important piece is that? Or could your system work with just like constraining it to choosing among a set of known to work phages? Like, what do you think about all that?
So I think first of all, it's really cool what they did. So like having like a system to make artificial phages which were never there before. So I mean that's really amazing. And I think also like parts of will be like used like hopefully there somehow. Currently, I think like we don't need to have like the whole phage always completely new. So if you say like you have a certain subset of really well functional phages, and then just exchanging like smaller parts like the tail fibers or certain anti-defense mechanisms. So imagine like you have like a kind of a breadboard which you like just putting stuff together to make the phage which like targets your pathogen or like the best. So for example, you see this is a certain CRISPR mechanism which this bacteria has. So you need this cassette for making the phage resistant to that. And if you see, I need this tail fiber for this bacterium, I pick this tail fiber out of my box. And you don't need all the proteins like newly. So I mean, for example, if you have a stable capsid part, maybe like having 3, 4, 5 different of these chassis, there you can, I think, go and like check these out and not having to make it completely from scratch, because I think it's also faster because like, they used rather small phage to do all their part. Not, I think, because of the limitation of the model, but like to make this DNA is like the limiting factor. And I think due to that, I think also like to having like this picking from like a larger set and then putting all the parts together, I think that would be also more like you can make it faster, and also like you don't have to say like, in 10 years we can synthesize 200 kb genomes in 2 hours or something, but you can already do like stitching parts together now. So I think that also quite helpful.
Yeah.
Just curious, how high a titer have you been able to achieve? That's my first question, and I have a follow-up.
And that varies a bit on the phage. So the smaller on average, the smaller the phage, the higher the titer is. So like with a really tiny phage, like an RNA phage, this MS2, we got like around 10 to the 13, 14. But I mean, it just has 4 genes, so it's like not too big. With like some protovirida, we like around 10 to the 12-ish. And then like 10 to the 11, 10 to the 10-ish on some other phages. So it varies a bit on the size, and that's because like the more proteins you need to produce, the more metabolic load you put on your system, and therefore like exhausting your ATP or like your fuel of your system a bit. So therefore you have to be a bit careful about the size.
And I guess the follow-up question to that is, I mean, do you know that, I assume, I mean, you're getting 10 to the 13th or 10 to the, with the smaller phages, you're getting that many phage particles. But I guess what I'm asking is, how many phage particles do you get that don't infect? Do you have any idea of whether you're getting a large number of phage particles that, like if you count phage particles under a microscope or something and try to figure out how many phage particles you have, and then you count PFUs, is it close to 1 to 1? I mean, I guess I'm partly asking because, people, I would be worried if you were getting like 10 to the 13th titer or phage, but you're actually getting 10 to the 15th phage particles, then you might start inducing immunity and, in the patient, and that would be bad. So I guess, anyway, that's the question. Do you have an idea when you have 10 to the 13th PFUs, do you have a roughly 10 to the 13th phage particles?
So we did some electron microscopy imaging of like conventionally produced phages and self-reproduced phages and compared these. And there are the numbers of like empty versus like filled phages look quite similar. So they were not like too much variations. I didn't check it for all the phages because
Sure.
but like for the test setting of when we initially had this question as well, it looked fairly similar actually.
Great. Do you have a way to monitor, like, because it sounds like the limiting factor is like ATP, polymerases, like energy and machines. Can you monitor in your reaction, like as those are getting depleted and then like add them back to keep the process going longer or to like extend it so there isn't a maximum?
So like we did some like extension experiments. So like the normal experiment is just to mix everything together, put it into the whatever incubation chamber and wait. But we also had a system where we have like a source outside which could you feed like back into the system. And there you can also reach like higher titers. Didn't work like infinitely. At one point there is some limit. We're not completely sure yet. We have some hypothesis what could be the limiting factor in the end, which cannot be depleted or is depleting. One part is that at one point the DNA is all gone, for example, because it should be all in the phage particles. But yeah, there are also some other factors which like doesn't give you like infinite amount of phage particles, but it's you can like increase the titer like around 2 logs roughly by like having like an outside source where you add like stuff to the system and also letting stuff out, right? That's the next thing, because like you get kind of generate, let's say, trash kind of, which you have to get rid of. So like changes of like some metabolites which are like would inhibit the system, and you have to also get rid of these, which is also can be done.
So I'm going to ask you an impractical, geeky biology question that I'm fascinated by. But if you put, say, lambda DNA or whatever, you pick your favorite temperate phage, and I guess for a lot of people it's lambda, but you put lambda into or some other temperate phage, does it think it should be making lysogens and repress and not make phage particles and establish repression, or does it make a bunch of phage particles and think that it's supposed to be going lytic?
So what we saw, like usually on default, it thinks it should be suppressed. So on average, they think they are in the lysogenic cycle, and
I see.
You have to do something to make it like think it's in the lytic cycle to make these phages. Every phage is individual, as I guess you also know.
Yeah.
But on average, we saw that trend rather.
Right. But I guess, So I guess, it would be really interesting to me if that's the case. Like, and then if you put in a phage that does, a lambda that has mutated for C1, the repressor gene, then it just goes fine. Or
so then if you like change something like we did some mutation and then the like repressing region, and then like we saw like then it worked again. So that was also cool. But you have to know like if it's an unknown phage. For the lambda phage, everyone knows everything.
Yeah.
There's a lot of in the papers, but some random phage you get like there you have to also check and see what's close by.
I wonder, like, people have been thinking about like what influences whether you make a lysogen or go lytic for years. And this is like one of the things that people still argue about. Phage geeks still argue about the lysis-lysogeny decision. And I wonder, like, if you, I don't know, like, just a simple experiment of if we have the extract at maximum concentration, does it favor the lytic pathway? And if you dilute it and get a less concentrated, I guess you probably are having more problems getting the phages to work. But it would be, it'd be really interesting to see like how the protein concentrations in the extract affect whether the in vitro you get lytic or lytic or lysogenic. I don't know exactly what you would learn, but this is one of these things that people have been arguing about since they discovered that lambda could do one or the other.
I think like maybe more important could be also like the state of the cells when we make the cell-free system. So we remove a lot of like the smaller metabolitic parts, but still like the composition of proteins is the composition of a happy cell, I would say. So it pretends a bit like, because my random guess would be if the cells are happy, then it can go on to the more lytic cycle, because then there's everything there to to replicate happily and make a lot of new phages. But for the cell-free system, it seems like, yeah. But if, as you said, maybe it's a protein content, yeah. But there we are a little bit limited in the ability to play around, because if you concentrate the cell-free system too much, you don't get like protein expression nicely anymore. So it's like there the degree of freedom to play around is a bit limited, I would say.
So I sort of naively have assumed during this conversation that you make your extracts from things that are basically growing in log phase, mid-log. You don't wait till they're saturated. Is that correct?
Yeah, so you can do even both. So I know also like other people who like harvest them in the stationary phase more or less. But we go like earlier, so for at least for us it works there the best. But it's also possible to go all kinds of different phases, even in the starvation phase, where like the bacteria start to eat each other. So
have you tried introducing a library of different phages to see which ones assemble better, or like a mutant library or something into your system?
yeah, so we tried like, for example, like tail fiber mutants to see like which is the best working one. So I mean it's quite straightforward, and then you can like check which one is working well. They have also different like options. I mean you can do it like genetically putting all these mixtures together, throw it in the cell free, and then just do a plug assay and pick some plugs and to see which are working or which are not. But you can also like do some assays with like microfluidics. So you can make like a lot of different phage particles in like a confined environment, and then like adding bacteria to see would it work or not, and then looking for the working phages. So that's also a possibility which you can use with the cell-free and some large-scale screening capabilities, because there you get like 10 to the 8, 9 particles you can sort within one experiment, even a bit more if you're more patient.
So I know that you guys want to expand toward phage therapy more, and we talked about your partners on the kit side and research partners, but do you also want clinical partners yet, or where are you kind of in that timeline?
So there right now we like about the indication, checking like which one would be the one to go for the first clinical trial. But I think it's still confidential. I don't know.
Okay, yeah. And yeah, just meaning like, can people help you, or like, do you want like some infectious disease doctors to reach out because they have patients or they want to start a clinical trial and need a source of phages? Like, can you be the manufacturer yet, or are we upstream of that, you think?
I think we're like maybe a little bit away from that. So if we have this machine ready, like this phage particle printer, I think then it would be like, yeah, like then I guess we like do also a lot of outreach to see like who wants to work together. So, but like we have to finish first all these building parts, doing like one step after the other.
Yeah. Yeah. And like in terms of the scale, we talked about like how many phages you can make in a batch, but can you like, I assume that's, well, actually I don't know if it was PFU per mil or PFU total, but can you make enough for like a clinical trial if you decided you were ready to, like physically enough volume and enough phages for that many doses?
Yeah, so like there are two parts to this question. So one is like to make the cell-free system like in larger quantities, and there currently we are with this partner in this tech transfer process and also like scaled up, so that we have like the initial starting part around like 10 liters. But for the reaction itself, so in this phage printer, the idea is to have rather like smaller volumes, so that you make more or less enough material for one patient.
Got it.
But in principle, also like you can. So we didn't make like a really large batch yet, because I mean that's also cost some money and everything. But it should be also possible to create like a larger batch if you say like you go for clinical phase 1 to a trial or something.
Okay. Yeah. So the scale is not the hang up. You're just kind of making sure it works reliably and that you can prove to the regulators that it's the same. And things like that. But otherwise, seems like a lot of stuff's figured out. Very cool. Do you have any other burning questions, Joe?
No, I think I've, I'm not shy about asking my questions. So I got them in. But it was fun talking to you.
Yeah, this is so cool. So what can the Phage community help you with, or any listeners? What are you kind of looking for? Are you guys hiring? Are you guys expanding? Do you need more partners to try your kits? Can people order the kits if they email you? How does that? What do you
I
need?
think like if you email Invitris, for sure, like there's a way to come to a conversation and check out like how to do something together. I think that's the case, and we're always happy to talk with people from the community, also to see if they have a cool or crazy project. I mean, like expanding the knowledge is always like important. And I hope like down the line also at one point like going more and more to clinicians, and also like having this in the direction of like a clinical trial, even if it's a little bit down the line yet for us. But then I think it would be also really interesting if someone like in a year or so hears this podcast, then maybe it could be
Yeah. And like all the exotic phages you could probably make. Like I bet a lot of people are limited in what company they might start, or like disease they might try to treat, because we just don't know a lot about how to make phages against that host. But if you are able to kind of expand that, I think that's really, like, this is just such a foundational technology to kind of go in so many directions. So I hope the phage community flocks to you to talk about how to do that. Very cool.
like at conferences. I'm also like happy to chat always. So.
Oh yeah, which conferences are you going to this year? Any phage ones?
I guess to the
Or other?
viruses of Microbes in Prague. I mean, it's not that close from. It's not that far from Munich, so.
Yeah, that's perfect. That one. We gotta advertise that. We don't have to, but we could. Viruses and Microbes. Are you going, Joe?
Planning to.
Yeah, in Prague, July. When is it? July something.
6th through 10th, I think.
6th to 10th. Okay, you can meet Kilian there and Joe.
I hope like we can meet each other.
We can re-meet.
Yeah.
Every 2 years we'll meet at Viruses of Microbes.
Yeah. Yeah. Well, yeah. Thank you so much, Kilian, for sharing. And I'm really happy we got to dive into the nitty gritty and figure out like physically what is happening. I feel like I have a good picture, and I just want to go order your kit now so I can ask all my phage questions. So expect some emails from me and maybe some more of our listeners.
Also, thanks to show the cell-free technology to a broader audience. So really happy to share all this.
Yeah.
It's a lot.
Great.
No problem. And do you have any papers out that people can look for? We'll put them in the show notes, but have you published on any of this?
Only some of the earlier stuff, like because it was like, when you start a company, then you're more into the patterns, and unfortunately don't have the time so much to
Yeah,
publish
totally.
everything.
Okay. Well,
Just
we'll
have to
put
take
some
the
early
title.
stuff.
Sorry, I don't know.
Oh, no, I'll look for it. All good.
Okay.
But good to know that it exists. And so if people want to kind of like find out more and read more. Cool. Well, thanks, Kilian. Have a great rest of your week. And good night. I guess it's night in Germany.
Have a good start into your day and all the best. Thanks a lot.
Yep. Thank you.
Bye-bye.
Bye.
Bye.